Good morning, everybody. Welcome at my lecture. Today's lecture will be about some introduction to the Biocybernetics. So here we have some information what is lecture about. So also this lecture is related to some oral exam questions. So we'll be talking about the information and its transmission and processing in biological systems. We'll be talking about biological signals, what is biological signals, how we can create biological signals, what is the definition of the biological signals. Also, we'll be talking about how we can make a recording of the biological signal, what is main difference between electric and non-electric biological signals, also what are the principles of the transducers for the recording of these non-electric signals. Also, I will provide some information about the monitoring and the telemetry. We'll be talking about the system, what is system, what is the definition of the system, something about the terminology, basic terminology, and also basic information about the structures. We'll be talking about the properties of dynamic systems and basic of organ control and regulations. All these informations are related to the oral exam questions, so you can pick up some of these questions during the oral exam. So please, everything what you will see on this presentation also you can be asked during the oral exam. Let's continue. When you will see the symbol on the slide, it means that it is good to know for the exam. It means that exactly this information we will be asking you during the oral exam. Okay, so please enjoy my lecture. It's up to you how you will enjoy it. Well, so we will start with the basic information. What is a cybernetic? Usually it came from the Latin or from the ancient Greek named cybernetic, which arose as independent science orientation after the Second World War, and this word Kybernetes mean something like a steersman. The originator of the cybernetic, or in general originator of cybernetic, is Norbert Wiener, who as a first discovered that it's good to join interdisciplinary subjects as a math, biology, chemistry together, and he as a first discovered a cybernetic could be a transdisciplinary approach for exploring of regulatory system and their structure and the properties, and also the systematic application of theoretical knowledge as a mathematics, chemistry, physics in study of biological system is, of course, biocybernetic. Okay, so that's a systematical application of all another subject into the discovery of biological system. So biocybernetic plays a major role in the system biology. When we are discovering or when we would like to integrate a different level of information, and when we would like to understand how biological system works. So basically, for what we need some interdisciplinary approach of this subject. For example, during the creation of some new materials, so the medical cybernetic applies knowledge for discovering or for developing of new material for the medicine, for example, micro and nanotechnology, because when you would like to create a new material, also you must have a knowledge from material science, something from the computer simulation, and so on and so on. So all subjects together will give you enough information, enough knowledge for creation of new materials. Also, direct application of medical cybernetic is for development of new diagnostic software and simulations, and also for development of new diagnostic tools and methods. Also, medical cybernetic is useful. Let's start from the beginning. At first, we will say something about the information. So the basic theory of the information is about the interaction of some signals or interaction of the information with the human body. So first of all, this is a happy human body, and our organism perceives the signals from external world and process this information as the signal. So it means that our body interacts with some information from surrounding, and some information inside of our body change a state of our body, and our body is making a response. So the organisms continually interact with the surrounding and with other systems, so it uses an information. Okay, so every time we have some interaction with our surrounding, so it means that we are using information. Let's continue. Information, there is a lot of possibilities about the transmission and processing of the information. So at the beginning, we can say some main or basic definition of the information. So, for example, it could be a sequence of symbols. So information can be sequence of symbols, then can be interpreted as a message, or it could be some state or behavior of the subject. So, for example, when you are describing some object, you are creating an information. Okay. So there are two types of information. First is analog, and second is digital. The analog information is, for example, sound, light, pressure, temperature. That's some kind of analog information. But anyway, our body or our brain is not able to understand the analog information. So all this information are later converted into the form of action potential. So it means into the form of digital information. So, but basically sound, light, pressure, temperature, and so on and so on, everything what our body is able to catch or is able to record it by the receptors is some kind of analog information, and then later we are able to convert this information into the digital form, which is action potential, for example. Okay, so another example, digital information, that's progressions of 1 and zeros, which carries an information about the, for example, sound or an image, and the basic unit of the information is one byte. Let's continue. Something about how the information can be transported, or a transmission of the information. So first of all, we must have a source of information. Okay, so something what is creating information. So on this example, you can see that we have a boy and he's creating an information. The information travels. In this example, that's flowing in the air, for example, and on the other side, we must have a user, receiver. There is a problem that information or the signal can be compromised by noise. Okay, so it means that information can be changed during the transmission by noise. So it could be, for example, if nerve is damaged, or when you are speaking loudly to the long distance, the final user is not able to understand everything because the information can be or may be compromised during the transmission. So the signals are transmitted to the receiver through a channel. Channel could be air, channel could be a cable or some conductor, and also channel could be a nerve, for example. What is very important is that we are able to protect this transmission counter noise, and we can use a code. So code is a rule for converting a piece of information into another form of representation. So for example, we'll make a code. So almost every information is coded. So, and when you have a code of the information, so it means that everything where you have the maximum, in this example, you have a number 1, and everywhere where you have a minimum, you have a 0. Okay, so in this example, you made a coding. And when you have a code, there is not possible to compromise the signal by noise. Okay, because the code is stable, and when you will create the code, you will send the code, and when the final user is able to open and understand the code, there will be no noise. What does it mean for the biological system? So usually everything in our body is coded for our brain, usually in the form of action potential. It means that our body again is not able to understand the analog signal, but our body is able to understand, or our brain is able to understand usually action potential. So the sensory pathways provide the information on the stimulus type, intensity, and the location. And final information, action potential, are coded by frequency, not by the amplitude. What does it mean? So here you can see a pressure applied on the pressure receptors, and here you can see how the action potential is created. So when you have a below threshold pressure applied, no action potential is created. But when you are increasing the pressure on the receptor, final action potential which is created has a higher and higher frequency, but amplitude is the same. Okay, as you can see here, amplitude is the same. So it means that the pressure is converted into the form of action potential and is coded by frequency, not by amplitude. Okay, so please remember that every signal in the human body is coded by frequency, but not by amplitude. Okay, frequency is changeable, amplitude is stable. Okay. Why we can hear or why we can see is that's about because we have an ear, and our ear works as a converter. So it means that our ear is able to convert vibration or analog signal or some mechanical vibration into the form of action potential. Okay, so as you know, we have a hair cells inside, and these hair cells are creating action potential, and this action potential is sent into our brain, and your brain tells you what you can hear. Okay, so this is a primer pathway for coding. Okay, now when we know what is information, we can say something more about the biosignals. So first of all, the carrier of the information is biological signal. Okay, so when you have information or when you are recording some biological signal, it means that you are recording information from human body. Of course, biological signals are generated in a live organism, because if the organism is not alive, so no biological signals is created, and it provides you information about body apparatus activity and the relationship with the surrounding. So it means that, for example, when you are recording the heart activity, also this heart activity can be affected by some relationship with another organs, or when you are recording brain activity, also there is some relationship with another organs. Biological signals are created by a vital manifestation of the organism. So it means that during the function of our organs. Okay, so during the function of our heart, you can record an electrocardiogram or electroencephalogram during the brain function, and so on and so on. Or you can create biological signals by stimulation from the external space. So it means that you will put a human body into some field, or for example, you can stimulate a human body by electrical current. That's an example what you are doing in this week at practical session A, when you are recording skin resistance. So you are creating biological signals by stimulation from the external space. Or magnetic field, ionizing radiation, so CT, MRI, sonar, X-ray. All these examples are, you are stimulating human body, and you are generating generating biological signals, but by external stimulation. Okay, so MRI signal is not created by human body itself, but you are stimulating the human body. Okay, let's continue. Okay, so here you can see some examples. Biological signals created by vital manifestation. So, for example, when we are talking about the movement, about the speed, about the pressure, that's an example of mechanical manifestation of biological signal. Type of sampling means how we can record this mechanical manifestation. So we are using mechanoelectrical transducers, and the direct diagnostic tool is, for example, spirometry, phonocardiography, tonometry, ergometry, and so on and so on. Okay, so this is a direct medical application. Another example: body temperature, surface temperature, infrared radiation. So you are using thermometer, liquid crystal detector, calorimeter, infrared detector, and so on and so on. And the final diagnostic tool is a thermometry, contact of thermography, or thermovision. So here you can see examples. Also, when we are talking about electrical properties as action potential, electrical activity of the muscle, electrical activity of a brain, for example, you are using electrodes. And here you can see example electrocardiography, electromyography, electroencephalography, reticulography, and so on and so on. So again, here you can see examples for electrical signals. Also, magnetic manifestation, quantum magnetometer, and here you can see examples. Also, we are able to record some internal environmental factor as a chemical factor, pH, partial pressure of oxygen, partial pressure of CO2. Also, we are using a special electrodes, and this is useful for monitoring of internal space. Okay, so also it's not necessary to remember all these examples, okay, but some of them, mainly active electrical properties and mainly mechanical manifestation. Okay, so it's up to you what you will remember, but it's good to remember some examples, not all of them. By stimulation from the external space, so for example, Röntgen radiation, type of modulation of these signals is a reduce of amount of X-rays, and for displaying we are using photographic plates, photographic films, or digital detectors, and also skiography, skioscopy is diagnostic tool, nuclear radiation, visible light, ultrasound, for example, ultrasound reflection or change in the frequency of ultrasound. We are using piezoelectric sensor. We told you something about what is piezoelectric sensor and how we can use, and also you had an experience with ultrasonography and with the Doppler manifestation during the previous practical sessions. Also, electrical current, that's about the electrical conductance on stimulation or about the electrical resistance. Displaying tool is about the electrodes, and we are using it for rheoplatismography or chronaximetry, and so on and so on. And final, electromagnetic field, we are using a type of modulation which is a resonance. We are using measurements coils, and diagnostic tool is MRI. Okay, let's continue. Okay, this is very important. Biological signals. Biological signals could be basically divided into the electrical and non-electrical part. Okay, so please remember this definition, or this how we can divide the biological signals. So electrical and non-electrical. Electrical signals are generated as a result of electrical actions. So usually action potential, and so on and so on. So, and the final form of electrical signal, which we are able to record, is in form of electrocardiography, electroencephalography, electromyography, electroneurography, and so on and so on. Okay, so please remember, electrical signals are generated as a result of electrical actions on a membrane. Non-electrical signals are created by movement, deflection, pressure, voltage, volume, or flowing change. In this meaning of voltage, it means a tonus, change of the tonus of something. And the basic or typical example of non-electrical signals is blood pressure or blood volume ejected by the heart, or muscle tonus, and so on and so on. Okay, non-electrical signal means has a no electrical part. It's not created by electrical manifestation, but it is created by movement, by pressure, and so on and so on. Okay, so please remember, typical example of non-electrical signal is blood pressure. Okay, here you can also find some examples of basic electrical signals or basic methods. So, for example, when you are using electrocardiography, this is a frequency band, frequency range, and also voltage. Electroencephalography, electromyography, electrogastrography, and so on and so on. Also, it's not necessary to remember all these examples, but I will recommend to remember these first 4 or 5 examples. Also, the blood pressure, blood flowing, respiratory rate is non-electrical signals, but of course for recording of the signals you must convert it into the form of electrical signals. Okay, so how we are able, or how we can make an acquisition and recording of electrical signals. Usually we are using the electrodes. Also there could be 2 groups of the electrodes. We have a polarized electrodes and non-polarized electrodes. Polarized electrodes, they have a changeable electrode potential during electrical current flowing. Typical examples are electrodes from the gold, silver, or copper, and they do not react with the tissue. Another example, non-polarized electrodes, they have a stable electrode potential, and typical example is silver chloride electrode, and so on and so on. Okay, so please remember that for recording of electrical signals, we are using polarized and non-polarized electrodes. Also, another type of electrodes depends on how big they are. We can use a surface electrodes, or needle electrodes, or micro electrodes. You have ever seen the surface electrodes? We used it for the recording of EMG when we did a lifting during practical session. So usually surface electrodes, in this example there's a sticker that's attached to the skin, and this electrode is able to record electrical activity under the tissue. Needle electrode is much more accurate because you can insert the needle directly into the muscle, and in this example these needles are useful for EMG recording. Also, if it is necessary, you can record electrical activity directly from the cell, and for this example you can use extremely small electrodes with very thin tip, and you can insert it directly into the cell without any damage. Okay, so also this for recording of electrical signals, that's very easy because it's enough when you have an electrode and also the amplifier. But if you need to record non-electrical signals, you must have a transducer, because there is a very important to convert non-electrical signal into the electrical signals. So for the processing and non-electrical signals, transducers are used. Usually transducer is a device that converts energy from one form to another form. So you will convert non-electrical signals into the electrical signals, because computer is able to understand just electrical signal, but not non-electrical signals. So usually here you can see some examples of transducer. It could be capacitive, inductive, piezoelectric transducer, and also some more examples. We'll be talking about the basics about the capacitive transducer, about the inductive transducer, and also there is also another type. So basically the capacitive transducer is useful for converting a pressure, so a non-electrical signal into the electrical form. Here you can see what is happening inside of this capacitive transducer. So there is a capacitor during practical session, we will tell you how the capacitor works. This capacitor works on the principle that you have a membrane here, and when the pressure is applied, the distance between the membrane and the static plate is changeable. And this changing of the distance will create a variable capacitance, and variable capacitance is converted into the different voltage, and the signal is created. Okay, so when the pressure is applied here, it will create a deflection of the membrane here, and it will change this distance between membrane and static plate, and this change of the distance will create a variable capacitance, and different voltage is created, and you have a signal. Another example, inductive transducer. Typical example of inductive transducer is a microphone. So again, sound waves are applied on the diaphragm. This diaphragm is moving, and this moving counter permanent magnet will create an electrical signals, and you have a converter, how to convert sound waves into the electrical signal. Here you can see the definitions, how the capacitor transducer works, how the inductive transducer works, and also we've been talking about the piezoelectric transducer during practical session with ultrasound. This is another form how we can convert the mechanical energy into the electrical signal, which is the voltage. Typical example was ultrasound probe. Okay, so here you can see how this how does it work with a patient. So we have a patient here, and you have a catheter inserted into the artery. The catheter is filled up with saline or with a physiological solution, and the blood pressure is conducted into the pressure of the saline. Here we have a transducer. So in this example, we are talking about this transducer here. Okay, so this transducer converts. There's a pressure transducer, converts a pressure from the physiological solution into the electrical signal, and the computer will show you pressure waves. Okay, when we know what is biological signals, how we can record biological signals, now we can say something about the biosensors. Biosensor is usually an electronic device able to detect the substances and produce the signals which can be measured. Okay, usually each biosensor consists from some biolayer. The biolayer can be made by enzymes, tissues, organelles, the whole cells. So that's a layer which is able to interact with some environment, or is able to absorb some information and change it into another form of information. So usually each biosensor has a biolayer, which is the main component of the biosensor. Also physical or chemical part, that's about the transducer, how this biolayer is able to convert this form of the signals into for example, into the non-electrical output. What does it mean? Here you can see, for example, we have a sample. It could be a blood, it could be a urine, or it could be a food, or it could be a solution, water, soil, and so on and so on. So from this sample, we will have the basic unit what we are going to detect. Okay, it could be a protein, it could be, for example, coronavirus, it could be some chemical component, it could be some antigen, and so on and so on. We have a biosensor, so it means that we will put this sample into the biosensor, and this biosensor has a sensitivity for this basic part, or has a sensitivity to the analyte, which could be a cells, enzyme, viruses, and so on and so on. And this biosensor will create a non-electrical signal, or is also able to create the electrical signal. Okay, so again, this sample will make a reaction on this biosensor, and this biosensor will create the information. Typical example for today is how we are making antigen COVID test. Okay, so again, we are using a nasopharyngeal swab methods. So you will collect a sample from the human body. Also, there is a very important process how you can how you will gather the information from this sample. And when your sample is ready, you will put this sample into the biosensor, and this biosensor has a sensitive layer. Okay, so when you're going back, this biosensor has a sensitive layer, and in this example, this biosensor will give you final information which is non-electrical signal. Okay, so because there is a chemical reaction, it showed you that if you are positive or if you are negative. So this is a typical example how we can convert non-electrical signals into the form which we can see, because we are not able to see a concentration of the virus in the sample, but we are able to see the mark which tells you that there is some threshold concentration of the virus, as in this example. Also here you can see another examples of very common biosensors. So if you are using a small smartwatch, that's an example also of the biosensor, because it's able to detect your blood pressure, your heart rate, and so on and so on. So the smartwatch, that's an example of biosensors. Also for pulse oximetry, we are using a biosensor. Thermometer, there's some kind of the biosensor because it converts the information about the temperature into the number. Blood pressure monitoring, weight scales, ECG, blood glucose level, and so on and so on. So these are all examples of biosensors. This is a fun historical information, very funny historical information. The oldest biosensor ever, the canary in the gauge. So usually in the history, the mining workers would carry down into the mine tunnels with them because during looking for some gold or silver, sometimes they open a space with huge CO concentration, but because in the history they didn't have the sensors for CO level. So they used the canaries. Okay, so if the canaries was happy and thinking, it means that there was a very low CO concentration. But if the canary died, it was because of CO concentration was too high. Okay, so this was the example of the oldest biosensor ever. Okay. Okay, now not every biological signal are ready for direct registration. Usually, we must amplify them because biological signals are very weak. So usually amplitude amplification is needed. As you can see, this is an example of biological signals at the beginning. Now, when we are talking about that, we also converted non-electrical signal into the electrical signal, but it is still not enough for direct registration, because usually we must amplify the amplitude. So here you can see amplitude is so small. With amplification, so we are using amplifier, and this amplifier will amplify the amplitude of the signal. Okay, but very important is that another characteristic of the signal is the same as a previous signal. Okay, so here you can see conservation of other characteristics, for example, the frequency range. So as you can see here and here, the frequency is the same, but just amplitude is changed. So amplitude is higher, or we can say the amplitude is amplified. Okay, that's very important because in the form of the signal or in the frequency, there is coded information. Okay, so when you will change another characteristic of the signal, you will change the information. Okay, so for amplification, it's very important to amplify just amplitude, not change a frequency. Also here you can see how we are able to make a processing of biological signal. So for example, we are able to analyze a frequency characteristic. Okay, so here you can see the signal. This is the amplitude modulation. So AM, maybe you know these abbreviations from the radio, AM and FM. AM means amplitude modulation. Here you can see frequency is the same, but amplitude is changeable. Or FM means frequency modulation. So here you can see amplitude is the same, but frequency is changeable. Okay, so AM and FM. Or also spectral analysis is possible. So you can analyze the spectra, and so on and so on. Frequency mapping. Each part, for example, of a brain creates some frequency, and you can add a color to each frequency range. So on this example, you can see a distribution of various different frequencies according to the color. Okay, so here you can see that each part of the brain has a different color because there is a different distribution of the frequency. And also it tells you something about what exactly is happening in each part, or maybe how does it work, or which part is right now in some process. Okay, so for example, when we are making some cognitive test, this part, because it's green, is not working, but right now this part has a higher activity, this part has a lower activity because different distribution of the frequencies, or different distribution of voltage, depends on what is the primary signal. Also, you can make a recording or transferring the biological signal to the distance. So we are talking about the monitoring and telemetry. That's usually about wireless data transfer. So you have some sensors on human body, and these sensors are sending signals without cables, without connectors, so wireless to the computer, to the phone, and so on and so on. So in the medicines, we are able to do EEG, so electroencephalography, electrocardiography, electromyography, blood pressure testing, and so on and so on. Or in the sport medicine, with monitoring and telemetry, we can record a distance, speed, and so on and so on. Okay, so again, here you can see you will just put the sensors and cables on your patient. If you remember when we did electromyography testing during the practical session, during the lifting of the load, we also used the telemetry. The unit was wireless connected with the computer. Okay, so you will put the sensors, and these sensors will send a signal into the cell phone, for example, or into the some unit, or and so on and so on. So this is a telemetry, so recording from the distance. How we can make how we can see a signal which has been recorded. So this is useful for monitoring of vital function. So here you can see all signals together. Device consists from several biosensors. This unit for vital recording. So here you can see electrical signals together with non-electrical signals. So usually electrical signals will be electrocardiograph or the electrical signals from the heart. Here you can see blood pressure, which is non-electrical signals, but was converted into the electrical form for displaying on this device. And also here you can see the maybe CO2 level or respiratory rate. Also here you can see the respiratory rate, heart rate, and so on and so on, body temperature. So usually this monitor for vital function displays electrical together with non-electrical signals. So everything what you can see here is electrical and non-electrical signals together. Okay, so another example, electroencephalography. There's a method for recording of electrical activity of the brain, usually with the surface electrodes, but also needle electrodes are possible. The final record or final information is about is electroencephalogram, which is record on a paper or record on a screen. It has 4 waves: alpha, beta, theta, and delta. And this electroencephalogram tells you something about brain activity. Is also useful for sleeping medicine. Is useful for distinguish epileptic seizures from other type of spells, or basically for check a brain activity. Electromyography. I hope so you remember electromyography, what we did. We used surface electrodes, but also needle electrodes are possible. And the final record is electromyogram, which displays you usually voltage. So please remember, during the electromyography recording, we are recording electrical voltage or muscle potentials, and it is useful for functional muscle examination, peripheral nerve damage, conduction damage, nerve to muscle, and so on and so on. Okay, so needle or surface electrodes. Final record is electromyogram. That's about electrical voltage and is useful for diagnosis. Also, I hope so you remember when we did the electroneurography, when we used a Viking device. So we did a nerve conduction study when we stimulated a median nerve. And with the surface electrode, we record a peak of response, and we check a time delay, we check a duration, and so on and so on. So this is also example of electrical signal. Another example is bioimpedance signal. Bioimpedance signal is created when you are stimulating a human body by alternating electrical current. So here you can see the electrical current is flowing to the human body, and usually the scanned voltage amplitude depends on the tissue density, tissue resistance, and this voltage amplitude is changeable, depends on how much you have water in your skin, how much body fat you have, and so on and so on, blood volume, muscle quality, and so on and so on. So you can use this example for checking of the body composition. Okay. Another example, phonocardiography or phonocardiogram, bioacoustical signal. Usually, a lot of physiological phenomena are involved in the sound generation. So, for example, you can hear your heart, you can hear your lungs, you can hear, for example, the air in your trachea. In this example, you can use a phonocardiogram, which provide you a real-time visualization of the patient heart. So the sound is converted into the form of picture, and you can see your heart function, and you can do analysis. Another example is biomagnetic signal. Biomagnetic signals is created during some local ion currents. So usually the action potential in the nerve creates a weak magnetic field around, and we are able to record this magnetic field. Okay, so for example, every organ creates a magnetic field, usually with very low intensities, something around 10 to minus 13 Teslas. So it is in picotesla, so brain, heart. So each organ creates a magnetic field, and you can record this magnetic field. So as you can see here, this is a magnetic mapping of the brain, or for example, recording of baby heart. You can use a magnetic field. You can record the magnetic field which is creating around the heart. Or here you can see the distribution of the magnetic field in the chest, and also it tells you something about the heart function, and so on and so on. Final example, biomechanical signal. Typical, that's a blood pressure. So biomagnetic signal is a biomechanical signal is about the blood pressure, make force applied to the artery wall. And with a suitable sensor, you can convert this mechanical signal into the electrical signal, and you will see it on the monitor as a blood pressure. Okay, so please remember, blood pressure, it's a typical example of biomechanical signal, or that's a mechanical manifestation. Okay, let's say something about systems. So now when we know what is information, how we can record the information, how we can make a process of the information, now we can say something in the general about the cell, or about the system. We'll be talking about something about the system, what is system, and what is the structure of the system. So basically, the system could be divided into the isolated, closed, and open system. Isolated system means there is no interaction with the surrounding, so no change of energy, no change of matter, so there is no interaction with the surrounding. Closed system means that there is a change just of energy. And the open system means the also energy and also matter can be changed. Typical example is a cell. And also depends on what is a behavior of the system in the time. We can have a static system, it means no change in the time, and the dynamic system is very changeable during the time. Okay, so in this example, a basic cell, biological structure, a cell is a dynamic and open system. Dynamic means because cell is growing, cells is older and older, or cells is dying. So it means that that's a dynamic, changeable system. And the open system is because plasma cell membrane is able to transport the substances, so has a permeability. So it means that there's an open system. Okay, so here you can see examples of isolated system. In this example, isolated system is a flask. For a certain time, flask works as isolated system because when you have a hot tea, you will have a volume and also temperature of the tea for the certain time. So it means there is a no change of mass, no change of energy, okay, but just for short time. Okay, a closed system could be a cup with a lid, as in this example. There is a no change of matter, so it means that, for example, your tea will stay in the cup, but there is a change of energy because after some time the tea is a little bit colder. Okay, so this is a closed system, and the open system is the cell. Okay, because there is cell has a permeability, so there is a transport of energy, transport of substances. Another examples of the system. So it doesn't mean the system is only biological, but it could be also technical. For example, a motor engine, also electrical circuit, or the algorithm. Okay, so when we are looking for a diagnosis, or when you are setting up diagnosis or making some processes, you are creating the algorithms, which is some example of the system. Also, a cell, a brain, but also a metabolism or relationship between each part. That's a system. Also, there could be ecosystem or economical system, tax system, and so on and so on. So the dynamic system is a system that's changed over the time, and its current state depends on the previous state and external stimuli. Okay, so please remember, dynamic system is very changeable during the time, and usually each dynamic system depends on what is input information into the system, and it is related to the output system, because inside of the system there is a transform, process, information, and so on and so on. So basic features of dynamic system include amplification or attenuation of the signal. So usually in the dynamical system, the information is changed inside. So for example, the cell will receive the signal, and the signal is in process inside of the cell, or it could be a time dilation, signal filtration, and so on and so on. So here you can see this will be dynamical system. For example, a cell, you have an input signal, and the information is in process inside of the system. So it means there is a transformation, amplification, attenuation. So signal is a change, and after these processes, the cell will make an output. Okay, so also each systems they can communicate between each other. And also they can make a control and regulation. So control and regulation are processes that are effectively maintained by the dynamic systems. For example, direct control, that's a relationship between physician and the nurse. Okay. Also, there could be autonomic response control. That's a work of medical team with a different level of management. That's an example of autonomous response. And the final manifestation of the signal regulation is the feedback. Okay. So here you can see the processing of the information. So when you will touch something hot, your finger or your thermoreceptor will detect the hot temperature. The afferent information goes to the brain or goes to the spinal cord, and after the processing of this information, it will send you a feedback into the muscle, and muscle as an effector will make an action. Okay, so this is an example how we are working with the information. So the basic organ and control regulation, that's about the feedback. Usually it could be without feedback. Without feedback means there is no reaction, or with a feedback, positive or negative reaction. Result of the system regulation is very important because of homeostasis. So here you can see example of without feedback, or when we are talking about the system control, if there is no reaction. Okay, so without feedback. So we have a system, for example, a cell. This cell is receiving input information, and the output information is almost the same as the input. So it means that output information has no effect on input information. It means, for example, adaptation of an organism to the environment, sensation of warm or cold. Okay, so it means that no reaction. So output signal does not react upon the control center. Another very important example is positive feedback. Usually positive feedback is useful for stimulation of physiological processes. Positive feedback is a cyclic process that can continues to amplify the body response to a stimulus. So, for example, we have an input, our system will create an output, and this output will amplify the input information. Typical example is fast auto-excitation of the elements of endocrine system or nervous system during the growing development of the organs. On this example, you can see a birth is an example of positive feedback. Okay, so when baby is growing, baby make an action or stretch the receptors activating the uterus. It will send information into the brain, some brain signals for release of oxytocin, and this oxytocin will create another contraction of the muscles, and so on and so on. So this is like cycle process, and when the contraction gets stronger until baby is delivered. Okay, so this is an example of positive feedback. Another example is control of blood clotting. So, for example, when we have a bleeding, this bleeding will call or will interact the platelets, and this platelets will send another signals for another platelets until you have more and more platelets, and you will stop the bleeding. So this is positive feedback. Very easy example is flock behavior. When the number of individuals which are running create a general panic, and more individuals are running, general panic is growing. So this is an example of positive feedback. Negative feedback is when you need to slow down or completely stop physiological processes. Usually also it is very important for homeostasis. So, for example, input will create an output, and this output will slow down or stop the input information. So, typical example is a regulation of blood pressure, pH value, or regulation of temperature. For example, when your body temperature is increasing, it will start a process of cooling, so you will activate a sweating mechanism, and the sweating mechanism has an effect that body temperature is decreasing. So this is a negative feedback. Or when your body temperature is decreasing, it will activate the shivering, so action of your muscle for produce heat, and the final result will be that your body temperature is increasing. So this is a negative feedback, okay? Because the main result of this negative feedback is stop some process or to slow down, cool down the process. In this example, you must decrease body temperature or increase body temperature. Okay, some information about the modeling of biological systems. So how we can make a study of biological systems, or how we can make a models. So usually the modeling in a medicine is a method of study of system properties, structures, behavior, and usually the model is based on a change of original to the similar object model. So here you can see typical example or typical model of a dog. So is very simplified representation of the of a reality. Okay, so the model is a simplified representation of the reality. Why we need to make a models, or why we are making the modeling? The modeling is for study of some processes, and useful in human medicine, where the research of some diseases is complicated or sometimes impossible. Also, you can also study a processing did by models. That's a simulation. Okay, the process generated by a model. Here you can see the difference between model and simulations. So this is a static model. And when you are making simulation, it means that you are modeling a processes which your model is able to do. So moving or any process, there's a simulation, and model is static. Model can be alive, usually laboratory animals, rabbits, or microorganisms, viruses, bacteria, and so on and so on. So that's a laboratory model for studying of some processes, or lifeless chemical structure, 3D computer simulation, and so on and so on. So for example, at our simulation center, you can find the models like Harvey, Metiman, and also a lot of another models. For example, Harvey is cardiopulmonary auscultation simulator, and it enables an auscultation of 30 cardiological findings. So this is a model. Also, Metiman, pre-hospital whole body patient simulator. And it's useful for control simulator with a complete patient monitor, and so on and so on, and so on and so on. So you can make a training on this model. And what is very interesting and also very important to understand, that on this model you can make a repetition of some simulations. You can make a training. You can make a mistakes. And then later you can correct your mistakes. So you can make a training as on original patient, which is not possible in the hospital to make a training on some patient. But here you can make a training, you can make a mistakes, and then you can correct your mistakes. Also, today are very popular models from the 3D printer. So when you have enough informations like pictures. And so on and so on, you can make a 3D picture. So this is a 3D model of pathological heart. Here you can see a wrong position of the arteries and veins. This is useful for training before the surgery. And also here you can see, you can make a model, and you can make a model of some part of the human body. Okay, also there is also possibility of PC model or network simulations. So this is example of brainstem PC model of neural network, where this part representing the population of the neurons. And these lines are representing interactions or relationship between each part of the neural population. some information about the computers in the medicine, but I think this is no more a question for oral exam, this computer in the medicine. But anyway, we can use it for administration, medical records, drug prescription history, medical reports, and so on and so on, and also for the diagnosis. Here you can see typical example, computer surgery, Da Vinci. It uses 2D or 3D images. Also patient monitoring. Finally, you are still recording electrical, non-electrical signal for patient of the patient body, and also then when you have a final information, you can make a reaction. Okay, so here you can see the final representation, how we can use a model, how we can use a biosignals. So this is a Da Vinci for robotic surgery, and there you are using electrical, non-electrical signals, simulation, modeling, everything together, and you have a wonderful surgery tool. Okay, that's all about this lecture. Thank you for your attention, and if you have a question, please, you can ask me. Still we have some time. I'm going to the computer to check if you have some questions. Okay, guys. So if you have questions, write me into my email, but also you can write right now into the MS Teams or into the YouTube. If no more questions, thank you very much for your attention. I hope you enjoyed my lecture, and see you later. Bye bye.